We will wait for 5 more minutes for other people to join and then we can start. Yes.
हेलो
Yes.
शिवानी।
Yes.
जी मैं डाउट कि जो मेरी असाइनमेंट है ना, तो
Okay.
उसमें से मेरा 2 असाइनमेंट है ना छूट गया है।
अच्छा।
और मैं ना नानो टेक्नोलॉजी कोर्स लिया है मैंने और 4 वीक का है वो। तो
अच्छा।
उसमें से 25% निकालेंगे तो कुछ इश्यू नहीं होगा ना? क्योंकि 2 छूट
अ,
गया है और 4 वीक का है।
अच्छा, सो 3 असाइनमेंट दिया गया है आपका, 2 असाइनमेंट छूट गया है उसमें से।
टोटल 4 असाइनमेंट है उसमें।
हाँ, तो 4 असाइनमेंट में से 2 छूट गया है।
हाँ, हाँ।
अच्छा, तो देखिए, आई थिंक हर एक असाइनमेंट के लिए मार्क्स तो होता है, तो अगर छूट गया है तो शायद उसमें मार्क्स तो कुछ घटेंगे। बट फिर भी आप एक बार मेल कर सकते हो। तो लाइक मैं पीएमआरएफ टीए हूँ। तो एक और टीए है एनपीटेल का, तो दैट पर्सन विल बी बेटर टू आंसर योर क्वेश्चन कि कितना मार्क्स कटेगा या फिर कुछ एक्स्ट्रा कुछ मिलेगा कि नहीं, टाइम टू सबमिट। ठीक है।
ओके मैम।
एक बार मेल कर दीजिए, आपके पास मेल आईडी है?
उनका मेल आईडी तो नहीं है मेरे पास। आप
तो
मे
आप
बी।
क्वेरी क्वेरी रेज कर सकते हो ना, आपको आपको आपको
हाँ,
जो अगर कुछ क्वेश्चन हो तो वहां पे रेज कीजिए, वहां पे आंसर मिल जाएगा आपको।
ओके मैम, थैंक यू।
हाँ हाँ, मैं बता दूंगी। आई नो द टीए, तो मैं उनको बता दूंगी आपके क्वेरी
हाँ, चलेगा।
देखने के लिए।
थैंक यू मैम।
हाँ हाँ, वेलकम। So shall we start today's session?
Yes, ma'am.
Okay, and how is your learning going so far? Are you learning new things in this course? You learned anything new in this course, or you already knew what is being taught? Yes, good evening. So basically, I think you guys were not there in the first live session. So I conducted one live session already, and in that live session, I talked about some characterization techniques in a little bit more detail, which had been taught in the first week of the course. So basically, I think if you guys have received a mail, then you might have got a Google Drive link. So that drive has the presentation that I discussed basically. And I whatever session was conducted has been uploaded on a YouTube channel with the same name as the course, Biomedical Nanotechnology. So if you want, you can go through. It's a 2 hour lecture. So if you want, you can go through the lecture. And if you have any queries, you can ask them to me in the next session, which will be conducted tomorrow, same time, 4 to 6. So basically, today we'll be talking about what a little bit elaboration on what you have been taught in week 2, since we missed one class in between. So
Ma'am, there is a small doubt I would like to ask.
yes, yes,
Actually, I had enrolled in this course last year, but
okay,
I could not appear in the exam due to some reasons. And then
okay,
again, I enrolled this year.
okay,
So the thing is, I want to give exam and the all the assignments afresh. So
okay,
by mistake, I applied for the exam. They said that people who have not applied for the exam last year, they can apply this year also. So
okay,
I applied for the exam last year's exam, and I have got the date for the exam, and I am
Okay.
submitting the assignments now, and I want to take the exam for this session.
Okay.
So can it be done?
This also, I guess you have to raise a query because the actual TA of the NPTEL course has to answer this. Actually,
Actually, ma'am,
I don't
uh,
know all details about how it is being conducted. I'm only supposed to just conduct live sessions regarding any doubt you have regarding course material.
actually, ma'am, last year what happened? I'm a pediatric surgeon, so I'm not
Yeah.
free. Just for academic things, I've joined this course, so I
Okay.
could not
Okay.
take the exam last year. So I
I
thought it
understand. I
will.
understand. Now the thing is that, like my senior, he is the TA for this, so he will be able to answer this question, that whether you can appear or not. If you join in tomorrow's session, if I see you in tomorrow's session, I can ask my senior on your behalf and let you know tomorrow if you are there in tomorrow's session
Okay, okay, ma'am.
what he
Thank you.
says. Like, you want to, what exactly do you want me to ask
Ma'am,
the senior?
last year I enrolled, but I could not
You could
take the
not give the
exam.
exam. Yes, I understood that. And
Yeah,
then,
yeah. I want to finish the assignments this year, and then I
okay,
will be appearing in the exam this year.
okay. So,
So
did you
when it was told
submit all
to
the
apply,
assignments from the beginning in the time
yeah,
given
I have
time
been
frame?
submitting the assignments for this course for this
Okay,
year, and
okay.
by mistake, actually, they said that you can enroll for the exam when it was last year's session. So when I was enrolling for the exam, I enrolled for last year's session. This year also, they are going to take the
Oh,
exam.
okay, okay. So you accidentally enrolled for last year's session instead of this year.
Yeah, yeah.
Okay, okay. I'll ask my senior and let me see what he replies. Okay, and then I'll get back to you.
Okay, okay.
You
Thank
can
you.
drop your email ID also. I can send you an email if I do not find you in call in case you're busy. So
Yes, ma'am.
you can drop your email ID in the in call messages, and I will keep a note of your email ID.
Okay, ma'am.
Okay, okay. Thank you.
I have done that, ma'am.
Yeah, thank you. Any other questions? Okay, thank you. I'll keep a track of your email ID. Okay, so no further questions?
No, ma'am.
Okay, okay. Then we will start today's session. So basically, today we will be talking about, let me share. What's, okay, can you see the presentation?
Yes, yes, it is visible.
Yeah, okay, thank you. Uh, 1 second. Okay. So basically, today we will be talking about DNA nanotechnology, a little bit brief about DNA nanotechnology. And then we will be talking a little bit about nanopore sequencing. Like, you might have heard this term a little bit in the somebody's. Yeah, okay. So nanopore sequencing. So you might have already heard of this term. If you were going through the lectures, nanopore sequencing might have popped up in 2 or 3 presentations. So we'll be discussing a little bit about what this nanopore sequencing is and its application, how you can use it, nanopore sequencing. And finally, we'll be talking a little bit details about carbon nanotubes. Okay, so this is the overview of the presentation. So I'll be explaining in more details. So first, DNA nanotechnology. Can anybody tell me what you learned in DNA nanotechnology? What you learned in DNA nanotechnology? It's like a generalized question. You don't have to be very precise. You can say something wrong. It's not a problem. Like, what do you think DNA nanotechnology is? So, basically, I think you might have been taught already DNA nanotechnology, as in DNA origami, self assembly, and what else in DNA nanotechnology? Yes, nanopore sequencing. So basically today we'll be talking a little bit brief about DNA nanotechnology, how you can use DNA as a nanotechnology tool for various purposes. Most of it has been covered in the course details. We will just read a little bit more. So DNA nanotechnology, basically DNA has the property of self-assembly. So this is very useful in forming DNA origami. So origami means forming structures. So because of the self-assembling property of DNA, it helps us to form origami structures. Can anybody tell me what DNA origami is used for? What can it be used for, DNA origami? Any application of DNA origami? Yes, come again. For?
Drug delivery.
Yes, for drug delivery. Correct. So DNA origami can basically be used for drug delivery. You can also have DNA hydrogels. Then you can have signal amplification and deoxyribosomes. These are all self-assembled DNA structures that you can have. Then in with inorganic materials also you can combine DNA, and like for with gold nanoparticles you can synthesize gold nanoparticles, and then you can surface functionalize it with DNA and target it for imaging or for sensing. Then you have two-dimensional nanosheets. These also you can functionalize with DNA and can be used for sensing. Fluorescent nanomaterials, magnetic nanoparticles. So all of these inorganic materials you can functionalize with DNA and can be used for various purposes. Magnetic nanoparticles you can use functionalize with DNA, target to specific cells, and then you can then if the magnetic nanoparticles have some heating properties, you can induce heat and try to kill cancer cells. So you can target cancer cells using magnetic nanoparticles also. Okay. Fluorescent materials, I think you have already studied about fluorescent materials. Any two fluorescent materials you have studied already? Any two fluorescent nanomaterials that you have studied already? I think it was covered in week 3. Fluorescent nanomaterials.
Quantum dots.
Yes, quantum dots, and if you have to use for biological applications, then carbon dots. Quantum dots are made of heavy
Carbon.
metal. Yeah, exactly, right. Carbon dots, because quantum dots are made of heavy metals, so it could be toxic to cells. And carbon dots are biocompatible. They are non-toxic for cells. So usually if you have biological applications, you mostly go for carbon dots instead of quantum dots. Okay. What else? So nanomaterial types for DNA nanotechnology. So basically you understand how you can use DNA in nanotechnology. You can combine with a different nanomaterial. You can functionalize those nanomaterial with DNA, and then you can use it for different applications, be it for therapy, be it for bioimaging, be it for sensing, or be it for drug delivery. Like DNA origami can directly be used for drug delivery. We'll see an example of DNA origami. So this is an example where DNA nanotechnology is used for sensors. So what this paper has done is basically they have synthesized a quantum dot and then they have coated the quantum dot with streptavidin. So we know the streptavidin binds with biotin. So they have also synthesized a capture probe, which is biotin. So streptavidin will bind to biotin. And when you have your target DNA, that target DNA will basically bind to your biotin. And your reporter probe will bind to the target DNA. So this capture probe, target DNA and reporter probe together, it will form a sandwich. Did you understand? So when you're illuminating, we know that quantum dots are fluorescence materials. So basically the quantum dot they have synthesized, it has an excitation of 488 nanometer. So what happens is basically when you're exciting your quantum dot at 488 nanometer, the quantum dot will emit at 605 nanometer. The quantum dot will emit at the emission of the quantum dot will be at 605 nanometer. But you can see here it is written FRET. Do you know what is FRET? Do you know what is FRET? Anybody? If you do not know, let me know. I will explain what is FRET. FRET. What is FRET? So FRET is basically fluorescence resonance energy transfer. So what happens when you have two fluorophores? One fluorophore will be exciting at a different wavelength and emitting at a different wavelength. And the other fluorophore, if they are very close to each other. So this FRET phenomena will only happen when two fluorophores are very close to each other. That is, they have a distance of 10 nanometer or less. If the distance between the two fluorophores are more than 10 nanometer, then FRET phenomena cannot happen. So if two fluorophores are very close to each other, if two fluorophores are very close to each other, that is at a distance of 10 nanometer or less, then the excitation of one fluorophore will be excited at a different wavelength and it will give an emission at one wavelength. The second fluorophore's excitation wavelength will be the same as the emission wavelength of the first fluorophore. Do you understand any confusion in this? So, any confusion? You have two fluorophores less than either at a maximum distance of 10 nanometer or less. First fluorophore will be excited and it will emit at a different wavelength. Excitation wavelength and emission wavelengths are different. So it will be excited at a particular wavelength and it will emit at a particular wavelength. The second fluorophore's excitation, the second fluorophore's excitation wavelength is same as the emission wavelength of the first fluorophore. So as soon as the first fluorophore gives off it emits at a particular wavelength, that will be absorbed by the second fluorophore. And the second fluorophore will emit at a much farther away wavelength. Okay, so this is called fluorescence resonance energy transfer. Basically, from first fluorophore to the second fluorophore, your energy is transferred. So instead of getting two emissions, because you are having two fluorophores, so instead of getting two emission wavelengths, you will get only one emission wavelength. So this is known as FRET phenomena. Main criteria for FRET is that it has to be very close to each other. Okay. And there should be no quenchers. Like no other molecule should be present which is absorbing the emission of the second fluorophore. If that happens, then you cannot see any wavelength. Okay. So basically, there is a FRET phenomena happening in this quantum dot where when you're exciting it at 488 nanometer, the quantum dot is emitting at 605 nanometer. But when the target DNA is binding, the reporter probe is bound and the reporter probe has a fluorophore attached to it. This fluorophore will absorb the quantum dot emission and it will itself emit at 670 nanometer. This will happen via the FRET phenomena. This is how you can sense the target DNA. But if you consider a scenario where the target DNA is not present, so if the target DNA is not present, then this sandwich will not form. So the reporter probe will not bind to the capture probe without the target DNA. As a result, if you are exciting your quantum dot, then your emission will be at 605 nanometer itself in the absence of the target DNA. So this is how you can use DNA nanotechnology for sensor application. This is one such example. Do you understand? Any queries? Any questions? Do you want me to repeat anything? Did you understand?
Yes.
Okay, thank you. So next we go for DNA origami. What is the application of DNA origami in drug delivery? So here this is another example. In this paper what they have done, they have taken M13 phage DNA and then these staple strands are basically helper strands. Okay, so these are basically helper strands. So with this M13 phage DNA and with these helper strands, they are self assembling into they're annealing and self assembling to form different kinds of origami structures, as you can see here represented. One is a triangle DNA origami, the other is a square DNA origami, and another is a tube DNA origami. So basically what is happening, it is forming three different structures. So what they did, they saw what is the retention of these origami structures, like what is the accumulation of these origami structures, what is the what is the accumulation of these origami structures in tumor cells. When they conducted the experiment, they found that this triangle DNA origami was mostly accumulated and retained in tumor cells. As a result, this was further used for drug delivery. Then they selected this triangle DNA origami. This DNA origami will have some, since this is a DNA, so it will have Watson-Crick base pairs, right? So dox means doxorubicin. So this is an anti-cancer drug, doxorubicin. They loaded this anti-cancer drug into this triangle DNA origami. This doxorubicin will intercalate into the DNA via the Watson-Crick base pairing. Okay, so it will intercalate between DNA strands. So that is how the loading of the drug happens in this DNA origami. And then they are injecting this setup into the mouse. The mouse is already having a tumor. So they're injecting it and then they're checking that what is the effect on the tumor. So how is the drug released? So it is proposed that the drug will be released, that since it's a DNA, so if you partially digest the DNA, it will release the drug. So this DNA doxorubicin loaded DNA origami. This was retained in the tumor cells via the EPR effect. I'm sure you have been taught what is the EPR effect. So via the EPR effect, it accumulated in the tumor cells, and then they were partially digested so that the doxorubicin can be released. This partial digestion is based on pH. Okay, so based changing the pH will partially digest the DNA and release the doxorubicin. As such, the doxorubicin will act on the tumor cells and kill it. So this is kind of like targeted therapy because it will only go and accumulate in the cancer cells. So this is kind of like targeted therapy. However, you can notice that you are not using any kind of ligand to target specifically to a particular cancer cell. Any questions?
No.
Okay. So next we talk about DNA nanopore sequencing. So what is nanopore? What is a nanopore? So nanopore is basically a very small hole through and through this small hole electric current flows. So if you have some kind of analyte which passes through this hole, so the electric current that will flow through this hole, that will be interrupted or disrupted. So each analyte, depending on its shape and size, will block current in a different way. As you can see in this picture, that when the small analyte is passing, you're getting a different signal. When you're seeing this blue analyte passing through the pore, you are seeing a different signal. And when you see this big analyte, which cannot pass through the pore, but it is still disrupting the electric current, you can see a different signal. So all these signals cumulatively are used to identify the analyte which is passing. So this is what is known as nanopore sequencing. Usually the analyte which is used for sequencing is DNA or RNA. You can also use protein, but it should be very small protein. It should fit, basically pass through the hole. Very big proteins will not be able to pass. So we will also see some examples of some transmembrane proteins which could be used for nanopore sequencing. So what is the basic principle for detection and base identification? So like I said, this nanopore sequencing, this is basically used for DNA and RNA sequencing. So nanopore sequencing uses electrophoresis to transport an unknown sample. A nanopore system always contains an electrolytic solution. When a constant electric field is applied, an electric current can be observed in the system. So basically your nanopore system has an electrolytic solution. So on application of an electric field, an electric current will be generated. The magnitude of the electric current density across a nanopore surface depends on the nanopore's dimensions and the composition of DNA or RNA that is occupying the nanopore. So like I said, when a DNA or RNA is passing through the nanopore, depending on its dimensions and composition, the current density will vary. Magnitude of the electric current density will vary, which will help you in identifying each base. So basically, you can identify each base in DNA or RNA, like which base is passing through the nanopore. That is how nanopore sequencing works. So sequencing is made possible because when close enough to nanopores, samples cause characteristic changes in electric current density across nanopore surfaces. The total charge flowing through a nanopore channel is equal to the surface integral of electric current density flux across the nanopore unit normal surfaces between times T1 and T2. So when the nanopore is of molecular dimensions, passage of molecules, for example, DNA, it causes interruptions of the current level leading to a signal. So basically signal like this is generated. Like I said, when each base will pass through the nanopore, each base will generate a signal and it will generate different kinds of signal. So that is the principle for That is the principle for nanopore sequencing. So this is an example of how DNA is detected using nanopores. As you can see, this is already a marketed product. So mini ion, it is called a mini ion. It works on Oxford nanopore sequencing technology. So in this mini ion, you have a flow cell. In this flow cell, as you can see, you have a application specific integration circuit. You have a sensor chip, which will sense, and an array of microscaffolds. Basically, your nanopores are in this microscaffolds. So this mini ion flow cell, it contains 2048 nanopores, which is used to sequence DNA or RNA. Okay, so when the ionic current passes through the nanopore, because a constant voltage is applied across the membrane, where the trans side is positively charged. Okay, so this side is positively, this trans side is positively charged, and your double-stranded DNA, basically, there has to be some helicase activity, because you need to unwind the DNA. Only single-stranded DNA can pass. So DNA has to be single-stranded to pass through. So there will be some helicase activity here, which will unwind the DNA, and one strand of the DNA will pass through the nanopore, and depending on how each base interacts with the nanopore, it will generate different kinds of signals. So under the control of a motor protein, a double-stranded DNA molecule, it is first unwound. Like I said, some helicase activity is needed. Then what will happen? The single-stranded DNA with a negative charge is ratcheted through the nanopore. Why? Because this is negatively charged, and you, like said before, that a voltage was generated in which the trans side was positive. So this is positive and the DNA is negative. So it will pull the DNA through the nanopore. And with each interaction, with each base interaction, there will be difference in which each base interaction, there will be difference in current density, and each nucleotide will be then sequenced. Any questions so far?
No.
Okay, thank you. So what are the types of nanopores? What kind of nanopores that you can use? So nanopore technology, basically it is broadly divided into two categories. One is a biological nanopore, another is a solid state nanopore. Currently, most recently, hybrid nanopores, they have been proposed because it it has the advantage of both biological as well as solid state nanopores. So some example of biological nanopores is these are also transmembrane protein channels, and they're usually inserted into a substrate such as planar lipid bilayers, liposomes, or other polymer films. So first example is alpha hemolysin. As you can see from the structure here, it has a tube-like structure through which your DNA or RNA can pass through. So this alpha hemolysin, this is an exotoxin which is secreted by Staphylococcus aureus, which is secreted by Staphylococcus aureus. And as you can see from the shape of the protein, it's a mushroom shaped heptameric protein. Heptameric means it has 7 units. So heptameric protein, and its limited pore size is 1.4 nanometer. So pore size is limited. Anything with a diameter more than 1.4 nanometer will not pass through this alpha hemolysin. Another protein, biological nanopore protein, is Mycobacterium smegmatis A, MSPA. This also, as you can see, has a structure very similar to the alpha hemolysin. However, you can see that alpha hemolysin, this is like straight at the end, and the MSPA, you can see this is a little bit tapering at the bottom. So this is also very powerful nanopore for reading information from 4 nucleotides simultaneously. So this the channel of MSP octamer is 1 nanometer. So it is even more fine in sequencing compared to alpha hemolysin, because the diameter is even less. So this tapering end is basically 1 nanometer. This tapering end is basically 1 nanometer in diameter, so which is relatively small and narrow compared to that of alpha hemolysin. So as a result, spatial resolution of single stranded DNA sequencing increases. So if you choose this MSPA as a nanopore compared to alpha hemolysin, then it will then this one is a better sequencer compared to alpha hemolysin. Okay. Then you have solid state nanopores, the second type. So this is something you artificially synthesize. So it's not a nanopore. You artificially synthesize and make a pore. So this is made of silicon compound membranes like silicon nitrides. As you can see in this picture, they've made a pore. More recently, the use of graphene as a material for solid state nanopore sensing has been explored. So basically, they're forming a small nanopore diameter and passing the single stranded DNA or RNA through it. And when the DNA or RNA is not there, then the electrons will flow. But when the DNA or RNA is there, depending on what kind of nucleotide base is present, the flow of electrons will change. So that is how this sequencing works in solid state nanopores. Any questions so far? Where have you heard graphene? Graphene. You know what is graphene?
Carbon dots.
Carbon dots. Yes, you can form carbon dots with graphene because it has carbon. But mostly graphene is used to form carbon nanotubes.
Carbon nanotubes, yes.
Yes, carbon nanotubes. So this is one, like the reason to discuss about nanopore is that this nanopore sequencing was very effective during the COVID-19 times. So this is an example of application of nanopore. You can see here that during the COVID times, it helped in detection of SARS-CoV-2. Basically, you know that gene sequencing takes a long time. This nanopore sequencing is so effective. It sequences very fast and it will give you result much faster compared to normal genome sequencer. So, and it's a handheld thing. It's like very small. It's like this much small. It's a handheld thing. So you can carry it to different places and you can collect samples and directly analyze there. Whereas you do not have to be in a lab and analyze. So these were some advantages for the nanopore sequencing. So basically what they did was they collected here, they were detecting SARS-CoV-2 virions in wastewater in a defined catchment area. So basically they wanted to see the level of SARS-CoV-2 infection in wastewater. So they collected samples from wastewater treatment plants and then in sterile tubes, and then they concentrated the virions. Concentrated basically centrifuged the samples. Then through the supernatant and with the concentrated sample, they lysed the virions, extracted, purified the RNA. We know that RNA is not a stable genetic material. So immediately after RNA purification, they reverse transcribed the RNA to complementary DNA, cDNA. They immediately formed a cDNA. Now this cDNA is much more stable. So this was again used, we know that during COVID times, real-time PCR was very helpful for detection. So this DNA was quantified using the viral titer was determined using real-time PCR, and then by NGS. This NGS is basically the nanopore next generation sequencing. So viral titer was determined using real-time PCR, and the sequence was determined using this nanopore technology. So this gave faster results, and you can run so many samples together. So much more easier and faster. Any question regarding the application?
No, ma'am.
Okay. So moving on, we will go to we will talk about carbon nanotubes. So before talking about carbon nanotubes, you need to know a little bit about fullerenes. What are fullerenes? So have you ever heard of the term fullerene?
No, I have not heard.
Okay. So fullerene is basically, this is also carbon based material. Basically, we were talking about graphene. So graphene comes from graphite. So graphite is basically stacked sheets of graphene. Graphene is a single sheet, and graphite is stacked. So basically, you have graphite, multiple sheets, stacked sheets of graphene. You exfoliate them, you get graphene. Now that graphene, if it is completely folded into a ball, then it forms fullerene. If you roll it, it will form carbon nanotubes. So fullerene is basically, you have different hybridized carbon atoms. Like in the PPT, you will see we are talking about C60 fullerene. So C60 fullerene basically has pentagons and it has some hexagons. So these pentagon and hexagon together, they make the fullerene by joining them together. You can see the picture here. This is the structure of fullerene. You can see this is the pentagon and you can see the hexagons here. So C60 fullerene.
Yes.
Yeah. So it is, as you can see, this shape basically resembles a soccer ball. So if you see a soccer ball with those black and white patches, you will see the shape is kind of like that. That is why it is named fullerene, because it is named on there is a building called Buckminster Fullerene. So it is the shape kind of looks like that. So that's why the name is fullerene. So it is like a soccer shaped ball. This molecule contains 60 carbon atoms. So total carbon atoms, this is completely a carbon nanomaterial. So it contains 60 carbon atoms and you can arranged as 12 pentagons and 20 hexagons. You can see the hexagons clearly here and you can see that they are joined by 12 pentagons. This is the pentagon. So the most striking property of the fullerene molecule. So apart from C60, there are other fullerenes also like C70, C80. They will be much bigger in size. C60 is the smallest fullerene. So most striking property is its high symmetry. It is very symmetrical. And there are, you can find two different kinds of bonds in C60. So the bond length in a pentagon. So this bond length in the pentagon is 1.54 angstrom, whereas the bond between pentagons, between pentagons, like between the pentagon and the hexagon, that is 1.4 angstrom. So you find two different kinds of bond length in fullerene. So fullerenes are used, has various applications. Mostly fullerenes are used as lubricant, as rocket fuel, and for drug delivery. So basically you can see it's like a hollow sphere. So inside is completely empty. So if you load some drugs inside this sphere, and then you functionalize the surface of this fullerene with some ligand, then it can go for targeted drug delivery. So this is a very nice material for drug delivery. So why we are talking about fullerene? Because basically fullerene, the reason we are talking about fullerene because when you're talking about carbon nanotubes, you will see that the mention of fullerene comes. So that's why I gave you a brief overview of fullerene, like what it is, so that when we're talking about carbon nanotubes, you will understand what is fullerene. So basically from this picture, as you can see in this picture, so what is graphene? So before talking about carbon nanotubes, it's very important to know about graphene also, because graphene is the main component of fullerene and nanotubes, carbon nanotubes. So here you can see graphene is an allotrope of carbon whose structure is one atom thick planar sheets of sp2 bonded. So graphene has sp2 bonded carbon atoms and they are densely packed in a honeycomb crystal lattice. So basically hexagons, honeycomb crystal lattice. So the term graphene was coined as a combination of graphite and the suffix "ene". So this rock you can see, this is the naturally. So graphite is something which you can naturally find in the environment. So this is a graphite rock. So this has structures. You can see the graphite structure here. So you can see multiple sheets. Each sheet is called graphene and multiple sheets together is called graphite. A mistake that people often make. So you should remember graphite is multiple graphene sheets and a single sheet is called graphene. So if you exfoliate, like I was saying, if you exfoliate these graphite sheets, then you will get graphene. If you roll the graphene sheet, you will get carbon nanotubes. And if you roll the graphene sheet such that both sides are open, then it is a carbon nanotube. And if you roll it in such a way that it is forming a full circle, like with the pentagons, you are attaching the graphene together, then it will form a fullerene. So basically the hexagons come from the graphene sheet and the pentagons are basically used so that it forms a complete circle to form the fullerene. But you can see in the picture that this nanotube, this is open-ended on both sides. But you can also form carbon nanotubes. Suppose you are using the carbon nanotube for drug delivery. So if you're using for drug delivery, if you're loading with some drug and both sides are open, your drug could like come out from the nanotube. So for that, you need to close the nanotube. So for closing the nanotube is when you need the fullerene. So you will take half of a fullerene and attach to this side and another half to this side to close your nanotube to form a like a capsule so that you can load your drug and then target it. Is it clear?
Yes.
Okay. Okay. So we talked about graphene. So now we'll talk about carbon nanotubes. So what are carbon nanotubes? So carbon nanotubes, basically they are CNTs are named based on their size. So since the diameter of a nanotube is in the order of a few nanometers, so basically carbon nanotubes, they are what kind of material? 0D, 1D, 2D, 3D. What kind of material?
Madam, can you repeat, please?
What kind of nanomaterial? Like there are different kinds of nanomaterial. 0D, 1D, 2D, 3D, one dimensional, two dimensional, three dimensional. 0D is zero dimensional. So where do you think carbon dots lie? Carbon nanotubes lie?
They are 3D,
No, not
2D.
3D. No, 1D.
Okay.
Carbon nanotubes are one dimensional. Why one dimensional? You will find the diameter of the carbon nanotubes are in nano range. So one dimensional nanomaterials will have only one out of the three axis XY three dimensions. Only one dimension will be in nano range. Two are in macro range, like more than
Yeah,
100
yeah.
nanometer.
Micro. Yes.
Yes.
Yeah,
So
remembered. Yeah.
carbon nanotubes, the diameter is the only one which is in the nano range. It could be very, very long. Carbon nanotubes could be very, very long in length. So lengthwise, it is in more than 100 nanometer. So that's why it is one dimensional. But if you think about graphene is two dimensional. Graphene is basically a sheet. So it is two dimensional. So from two dimensional material, you are rolling it, folding it to form a one dimensional material.
Okay. Yeah.
So, so it is written that they can be up to several millimeters in length. So it could be so big in length. The nanotubes stem in their quasi one-dimensional structure and the graphite-like arrangement of the carbon atoms in the shells. So basically CNT is rolling up a graphene sheet to form a tube. So you can see here, graphene sheet is there. You're rolling up the graphene sheet to form a carbon nanotube. So nanotubes are formed by rolling up a graphene sheet into a cylinder. So you can see it is forming a cylinder. And then, like I was saying, that if you have to close the cylinder, depending on your application, sometimes you do not need to close the cylinder. Like for example, when I think that example might have been given, carbon nanotubes can be used for improving resolution in AFM. Do you know how? Do you know how? Carbon nanotubes can be used for improving resolution in AFM. Basically, you have the AFM cantilever. So it's very fine. The cantilever tip is very fine. And the AFM works on three modes: contact mode, non-contact mode, and tapping mode. Usually,
Yes.
yeah. So usually you do not use the contact mode because it damages the cantilever. And the cantilever is very expensive. So, and it's like very, very small. Cantilever is very small. Because it is so small, you can understand that the cantilever images also that you will find online, they are same images. So that much small the cantilever is. The cantilever tip is very fine. So the cantilever tip, essentially, it is said that only one atom is there at the cantilever tip. It is that fine. So, but that also has some limitation. So mostly the tapping mode is used because it gives a good resolution. There is some compromise in the resolution. Contact mode, very good resolution, but it damages the cantilever as well as your sample. Non-contact mode resolution is very poor, but it does not damage your sample and the cantilever. And tapping mode is basically, this is a sample surface, it taps. So sometimes it will come in contact, it will tap. So it will give you a fairly good resolution, but again, and it will not damage your sample as well as your cantilever. But the problem is that it gives a fairly good resolution. So if you have to improve resolution, what they do is nowadays they propose using a carbon nanotube at the end of the cantilever tip. So if you use a carbon nanotube at the end of the cantilever tip, it increases your resolution very high. More finer. The cantilever tip is very fine. Then the carbon nanotube is there. So that makes the tip even finer. Plus carbon nanotubes have some metallic or semiconductor properties. So very easily it will, if there is any charge flow, it can detect very easily. It can transfer the charges very easily. So by the tapping mode, when they use the carbon nanotubes, it improves the resolution. I don't know whether this was mentioned in the NPTEL lecture classes or not, but I think it was mentioned once. Not in much detail, but a little bit mention about how you can use carbon nanotubes in AFM. AFM, atomic force microscopy, in case you do not know the short form. Okay.
Yes, got it.
Yeah. So basically, like I said, you will only form nanotubes like by rolling up the graphene sheet, and then you will close the cylinder by capping each end with half of a fullerene molecule. Fullerene molecule, like I said, it will form like a capsule. So this is the type of CNTs. I think you have already learned the type of CNTs. One is called a single wall carbon nanotube. Another is a multi-walled carbon nanotube. So single walled is basically one graphene sheet rolled to form a nanotube, and multi-walled is you have taken multiple graphene sheet and then you have rolled it. So you can see one graphene sheet into another. Okay. So do you know which one is easier to make, single wall or multi-walled?
Multi-wall?
Multi-walled, yes, correct. So naturally you will get multi-walled carbon nanotubes very easily. Single-walled carbon nanotubes are very difficult to synthesize. You need a lot of restrictions, lot of conditions to make single-walled carbon nanotubes. Okay. Carbon nanotubes, like I think you have already been told that it forms, like it's not biocompatible. Carbon nanotubes are not biocompatible. It does not dissolve in water. So for that, you need to do some kind of functionalization. So I guess it has already been discussed what kind of functionalization you can do. Covalent functionalization, covalent modifications, then endohedral functionalization, exohedral functionalization. So different kinds of functionalization you can do for solubilizing this carbon nanotube. So if you want to use your carbon nanotube for like biological purposes, it's a carbon based material, so it will not be toxic, but carbon nanotubes are toxic if inhaled. Do you remember the processes by which carbon nanotubes are synthesized? Processes by which carbon nanotubes are synthesized?
One is gas and probe. There is some gas and some probe.
One was laser ablation.
Yeah.
Arc discharge method and chemical vapor.
Yes. Arc discharge method, laser ablation, arc discharge method, and another one was chemical vapor deposition. Correct. So chemical vapor deposition is mostly used. So the main problem with chemical vapor deposition is that you have to be like carbon nanotubes. If you inhale it, it can be it can accumulate in your alveoli sac. So that can cause cancer. So and it's very fine. So it will be like soot or dust and you will just inhale it. So very dangerous. So whenever you're synthesizing carbon nanotubes, you are supposed to use masks and gloves and take proper precaution. You should not inhale the carbon nanotube, and it's like very light, so it will just fly. And also carbon nanotubes will also be naturally generated. Like if you burning that soot that is generated on burning, that also is carbon nanotubes. We do not realize it, but that soot is also carbon nanotubes. So those are precautions for synthesizing carbon nanotubes if you ever have to in the future. So there are another way you can classify carbon nanotubes. So what are the classification for that? So first is rolling of graphene. So rolling of graphene gives three basic types of carbon nanotubes. So you can roll a particular sheet in different directions. So based on the direction that you're rolling, you can get three different kinds of carbon nanotubes. One is an armchair, one is zigzag, and another is chiral. Okay, you can see the top view of armchair, zigzag, and chiral, and you can see that each one is different. So I'll tell you what this n equals to m and n0 and m these are. These are basically that when there is a graphene sheet, like I'll show you the sheet. So this is your graphene sheet. When you're rolling it, it has a vector component called the rolling vector or the chiral vector. So that vector has those n and m values. So depending on your n and m value, if your n and m value is equal, then your carbon nanotube will be armchaired. If your n, you only have an n value, there is no m value, then it will be zigzag. And similarly, if you have different n and m value, then it will be chiral. So depending on how you are rolling your carbon nanotubes, you will get different kinds of structures of carbon nanotubes. So these are also very important because these structures affect the properties of your carbon nanotube ultimately. Your conductive properties, your tensile properties, your material property, everything will be affected depending on what kind of structure forms. So types of carbon nanotube, again, like I was talking about, see, this is your graphene sheet, as you can see, and this C, you can see it is written C, this is your vector. So you can see the two parameters N and M here. So if you're starting from here and you're rolling straight where M is 0, then you will form zigzag. If you're rolling in this direction, then it will form chiral. And if you roll in this direction, then it will form armchair. So this direction, you can see here it is written theta. This theta is known as the chiral angle. So if your theta is 0, then it will form zigzag. If your theta is less than 30 degrees, it is more than 0 degree but less than 30 degrees, then it will form chiral nanotube. And if it is equal to 30 degrees, then it will form armchair. So you can understand how it is being rolled so that it is forming different structures. Any confusion, any queries?
No.
Okay. So this is another type of carbon nanotube. So this is basically like same zigzag, chiral, and armchair we are talking about. So here you can see these points. You can see that it represents semiconductor. These yellow points represent semiconductor properties and the red one represent metallic properties. So depending on the rolling, your carbon nanotubes could be semiconductor or metallic. Armchair is always metallic, as you can see here, that when you're rolling, you're getting all metallic properties. So armchair will always be metallic. Whereas for chiral and zigzag, there's a formula n minus m equals to 3q. So if this q value is an integer, then it is metallic. If it is not an integer, then it is a semiconductor. In all other cases, a semiconductor. Most of the time, zigzag and chiral are semiconductor. Most of the cases, very rarely it will be metallic. And armchair is always metallic in nature. So this is how you understand some properties of carbon nanotubes based on how it is folded. So this is an example. This is the last slide. So this is an example of application of CNTs in sensing. So what they have done in this work is basically they have taken they have taken a substrate which is ITO. Do you know what is ITO? ITO. So ITO is basically indium ITO. Anybody? Have you heard of ITO? It's mostly used as a platform. ITO?
Indium tin oxide.
Yeah. So ITO is indium tin oxide. It is basically a very indium tin oxide is basically a very transparent substrate. So it is very transparent. So ITO is basically used as a screen in televisions. It's very conductive. It's a conductive surface, but it's transparent. Transparent and conductive. So that's why it has applications in television as a screen. So this ITO, this is a very useful substrate because of its conductivity. And here you can see the carbon nanotube. So basically the carbon nanotube is the sensor. Okay, so you have the carbon nanotube. Then what they have used is they have taken carbon nanotube. This carbon nanotube, they have coated it with polydopamine. So this has become a polydopamine coated carbon nanotube. So as you can see here in the picture, polydopamine, this is functional, this is surface functionalization of your carbon nanotube. So what they have done is they have coated the carbon nanotube with polydopamine. As you can see here in this picture, this polydopamine has NH2 groups and OH groups. NH2 groups. So basically you need these NH2 groups for EDC NHS reaction. It is a conjugation reaction. So for conjugation purposes, this carbon nanotube was coated with polydopamine. This is a folic acid. This is folic acid. This yellow thing is folic acid. So what? Yeah. So what they have done is, using EDC NHS chemistry, they have conjugated the folic acid onto the polydopamine coated carbon nanotube. And then there are some empty spaces left. Those empty spaces were blocked using BSA nanoparticles. Okay. So the empty spaces that are left, that is blocked by BSA nanoparticles. Now what will happen? Your cancer cells, certain cancer cells, not all of them, they will express folate receptor. Like breast cancer, HeLa cells, they will express folate receptors. So these folate receptors will come and bind to your folic acid, which is functionalized onto the carbon nanotube. So what you're basically doing, you are ultimately, you have taken a platform and placed the carbon nanotube functionalized with folic acid, and then the cells are coming and attaching. When the cells come and attach, there will be a change in there will be electrochemical change in your carbon nanotube. This electrochemical change will be transferred to I2O, and then it will detect a change, like there will be a change in signal. Initially, when the cells do not bind, then a signal will be generated, and when the cells bind, there will be a change in signal. There will be a deflection. Here you can see they have done HeLa cells. It's not very clear, but it's written HeLa cells. So, this electrochemical change helps in detecting your cancer cells. Also, if you tag your like receptor or cells with some fluorescence tag, then you can also do fluorescence detection. Okay, so this is one application of CNT in sensing, how they are used in sensing. Any questions you have?
Yeah, I have written few questions.
Yes, please ask.
Yeah, I had a question that in the peptide ink,
Peptide ink, yes.
yeah, peptide ink works as an adhesive.
Peptide ink will work as an adhesive if you're adding some molecules which have adhesive properties.
No, but like regarding the peptide ink, it was said that if it is a kind of a what we say, a kind of a mold or
Hmm.
some kind of like that, so
Hmm.
that if you want to write, you say if I want to write ITM,
Hmm. Hmm. Hmm.
uh, IMT, yeah, MIT, and
Hmm.
so with that peptide ink, you will write MINT, then
As far as I remember, it had different properties. You had a scaffold, you had a spacer, like that
actually
for writing.
uh, I don't actually remember regarding that, but I have while watching the videos, I had written that uh,
Okay.
how to print protein ink. That was
How
also my question.
how to print protein ink? Okay, so
Yeah.
that's like I have not worked with protein ink, so
Okay.
I
Okay.
cannot tell you exactly how to. Like you want to know how to
Mm-hmm.
make protein ink.
No, no, means like what is the basic function of protein ink?
Oh, okay, okay. Basic function of protein ink. Okay. One second. Let me see protein ink. Peptide ink you were talking about, no?
Yes, peptide ink.
Okay.
Cell adhesion. Then my another question in regards to the same topic was cell adhesion to what? Like drug or gene?
Come again. Another question. I will be writing down your questions. Do not mind if I cannot answer your questions.
Yeah,
I will
definitely not.
address them
Yeah.
in tomorrow's class. I will find it out, the answer,
Yeah, yeah,
and then
sure.
I'll let you know. The first
Mm-hmm.
question was what you wanted to know.
Peptide ink, does peptide ink work as an adhesive? Like,
Does
and it
peptide
is
ink work as an adhesive? Okay, first and
then second is like cell adhesion,
hmm,
like it has a cell adhesion. Like if you write MIT,
hmm,
so then there is a cell adhesion.
hmm.
So cell adhesion is to what? Like drug or a gene?
Cell adhesion is to drug or gene. I don't think it's cell adhesion to drug or gene. I think it's the adhesion to the protein. I think it's a peptide ink. If your cells come and adhere, so maybe it's a kind of maybe it's a possibility. I'm not saying that that might be the case. I'm saying it's a possibility that it's a peptide ink, so you have peptides, so those peptides could target to bind to some ligand or something on the cell surface and then help it to adhere. That could
Actually,
be
I'm
a
not able to
possibility.
understand protein ink only. Like
Okay, okay.
for what is the basic function of protein ink? So if
Okay,
you
okay.
could,
So basic function
yeah,
of you want to know about basic function of protein ink
protein
and
ink
how
also.
it is being used.
Yes, yes, yes.
Okay,
That is what.
so for
Yeah.
you, then what I can do is for tomorrow's class, I can make a short presentation before on Protein Inc. and one application, like how they have used Protein Inc. for work.
Please, yeah, yeah,
Okay, one article where they have used
Mm-hmm.
Protein Inc. and I will try to explain it to that. I
Okay,
myself will also learn and I will try to explain it to you. And then if you have any further questions, let me know.
yeah.
Anything
Um,
else apart from Protein Inc?
yes, I have a series of questions, if you don't
No
mind.
problem. Just let me know. I will make slides accordingly.
Yeah. Why linker is made up of hydrophobic amino acids?
Okay. Why linker is made up of hydrophobic amino acids? Linker for
Actually, I have also written, and I have not written the why that question arised. I have not written
Oh,
that. Now,
okay, okay.
next time I will write
Okay,
it down
yeah.
that also.
The thing I was saying about peptiding, it has a
Mm-hmm.
ligand, anchor, and linker.
Yes, it
Yeah,
has ligand, anchor, and linker.
yeah.
Yes, yes,
So,
yes.
so the linker, yeah, linker is basically string of hydrophobic amino acids.
Yes, that is what.
Okay.
Yeah, yeah,
So why hydrophobic and not why not hydrophilic?
yeah, yes, 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 yes.
Okay, fine. That also then I will address in peptiding itself.
Okay.
So any other questions you have? Any other queries?
The um, what is okay? No, that is freeze drying. Okay, fine. Excess buffer solution. Actually, how does PEG coating degenerate and release the free drug?
How is PEG coating degenerate and release the free drug? PEG is
Hmm.
basically PEG, right?
Peg,
PEG is
yeah,
basically it's like a stealth technology. So,
yeah.
PEG coating, it's basically a polymer. So,
Hmm.
by action of,
Enzymes
yeah,
or any bodily condition?
yeah. Enzymes, it could be polymer, some difference in any kind of
Chemi-
pH, yeah,
chemical changes,
chemicals,
electrical
pH change,
changes.
electrical, any kind of changes can help degrade
Hmm.
PEG. It's a
Okay.
polymer, so it can remove the coating, it can degrade. Also, we have a lot of enzymes in our body that can also help degrade the polymer and help in
Okay.
release up the.
Okay. And cationic liposome complex also I wanted to understand.
Okay, cationic. What you wanted to know?
Liposome complex.
Cationic liposome complex. One second. Pharmaceutical. Okay. Cationic liposome. Cationic. Cationic liposome complex. I do not
Yes.
see a cationic liposome complex.
DNA cationic liposome complex.
DNA cationic liposomes complex. This was, did you write in which topic it was under?
Actually, I have not written the heading of the topic, and next time I'll do that.
Okay, okay.
Uh-huh.
So, structure.
I need to actually write in more detail because like this it won't. Okay,
Yeah,
fine.
just tell
Yeah,
me
and
the
the
topic
module
under,
name
yeah,
and
module under
yes,
which it
yes.
is.
Sure, sure. Next time I'll do that.
Okay, this one also
And
I will address DNA cationic liposome.
cationic liposome complex.
DNA cationic. Okay, I will look for this one also and I'll explain it to you.
Yeah, and two more questions. What is Cephardex and explain nano emulsion? These two questions.
What is Cephardex? Cephardex
Hmm.
is basically, have you heard of during column chromatography? Cephardex column beads.
Mm-hmm.
That's Cephardex. Cephardex is a
Okay.
material which you form beads for
Okay.
column chromatography. Cephardex.
Okay. Okay. Okay.
And
Fine.
another one,
Okay. And I wanted to understand more nano emulsion.
nano emulsion. Okay.
Yeah.
Nano emulsion was in lipid nanotechnology, right?
Yes. Yes. Yes.
Nano emulsions. So basically, what you did not understand in nano emulsions?
I don't know actually because I have not written in brief. Like I have written just short. Next time I will write in brief. But
Okay, so
I
basically,
have, yeah, I have written entire like explain nano emulsion.
okay,
Just like that in question
okay.
mark.
1 second, I will share. You can see this one. This one, can you see?
Yes, nano emulsions. Yeah.
Okay. You can see. Fine. So nano emulsions is basically, you can see it is written here, emulsions with droplet size in the nanometer scale. Okay. So basically you have a hydrophilic phase and a hydrophobic phase. So to
Yes.
form an emulsion, it has to be like emulsion is thermodynamically unstable system consisting of at least two immiscible liquid phases. So
Yes.
like emulsion will only form when two liquids do not mix with each other. So if they mix with each other, you cannot form emulsions. So that is why the two phases, one is a hydrophilic phase and one is a hydrophobic phase, but it is thermodynamically unstable. Okay. So this is they are saying that one of which is dispersed as globules. So globule you can understand. So if you have like water and you put oil in it, which one will form the globule?
Oil will form the
Oil will
globule.
form. So because oil is the oil is forming because oil is hydrophobic and hydrophilic phase is your water. So here in this case, if you take the example of water and oil, then your globule is oil, which is the dispersed phase, and in the other liquid phase, the continuous phase is your water, which is the hydrophilic phase. Did you understand?
Yes.
But your hydrophobic, your emulsion is unstable. If your emulsion is unstable, so it will deform. So if you put your hand in oil, like if you put water in oil, it will form different kinds of shapes, right?
Yes.
Okay, so it's unstable. If you hit it, maybe it will break into two. It will break into smaller particles.
Yes. Small
Yes,
particles.
so it is unstable. So that to stabilize it, you're using an emulsifying agent.
Okay.
Okay, so that is like, for example, surfactant like SDS. It forms the interfacial, so that will stabilize your emulsion. That is called nanoemulsion.
Okay.
Can you
Got
understand?
it. Yes, I got it.
Okay,
Yeah.
okay.
Thank you. Yeah, my questions end over here.
Okay,
I
okay.
had this
Okay.
much questions.
Still, can you? I will repeat some of your questions.
Yeah.
One was regarding a peptide ink.
Peptiding. Yeah.
Yeah. What questions you had? First is
Peptide.
why
Yeah.
using hydrophobic linker?
Yes. Yes. Why
Why
the
using
linkers are made
hydrophobic
up of
linker?
hydrophobic amino
Okay.
acids.
Then peptide ink, how it is used, like
Peptiding.
how it is useful,
Yes.
how cells are adhering to peptide ink,
Yes.
how it is
Yes.
useful in nanotechnology, how you can use it, and what else?
Other questions were answered like, the another one is DNA cationic liposome
Okay,
complex.
DNA cationic liposome complex. I did not find it. If you had written the module, maybe I could have addressed it right now. But I can't seem to find it. I will check DNA cationic.
Yeah, DNA cationic liposome complex. If you have some email ID, then I can
Fine.
provide
I
you
I
the
will.
details on them.
I'm typing my email ID. Okay. I've typed my email ID.
Yes.
DNA cationic. Okay. DNA cationic complexes. I think it will be in either DNA nanotechnology or lipid nanotechnology, but I can't find it in lipid nanotechnology.
I'll just I'll email you the details of it,
Okay, okay, okay.
where I found it.
Okay, okay. Is it no, no, no. Okay, fine. You
Yeah.
can tell me the details. I will look into it.
Yeah. And another question which I had that is IIT planning any workshops for the one who has taken the courses? Like if a 2-day workshop can be arranged.
2-day workshop for this biomedical nanotech?
Yes, yes.
No, no, no. I don't think so because NPTEL is usually online courses.
Yeah.
So I don't think any workshop like for you to come and learn for that purpose.
Yeah, a
Yeah,
workshop,
yeah, no, no,
yes.
no, no. Sorry, no. I don't think they.
Because Yeah, they had earlier, they had some programs of workshop. From now,
Usually,
they are not.
sometimes what happens, not through NPTEL. I don't think
Uh,
through NPTEL they will ever do such things. But usually, some workshop is there, but that is completely different. That is not related to NPTEL. So
okay,
some
fine.
workshops always they keep happening, but it will not be
Uh-huh.
related to NPTEL.
Okay, and if we want to be the part of those workshops, then
Then you have to keep track of what kind of workshops are happening, and then you have to
okay,
apply to like there will be some news. So
link
like,
or
yeah,
news, yeah.
link, or you have to fill up some forms, Google Doc forms. I think some kind of payment or something you have to do. Then your stay and everything will be arranged, and then you can come attend the workshop.
Okay, fine. Thank you for that.
Okay, okay. Tomorrow I'll be trying to address the questions that you asked. Thank you for asking them. I don't know how much I'll be able to answer, but I'll try my level best to give answers to your questions. And
Okay.
like anybody else have any other question?
Ma'am,
Yes.
will we have these sessions like before our exams also to have doubt clearing?
Currently no, because I missed 1 week class, so today that's why I'm conducting that class. Tomorrow is the scheduled day, Saturday, and next week, last Saturday, I'm having class. If you want any doubt clearing session, then we have to arrange one extra class where I'll not be presenting anything. But then what happens is that if you're asking me a question and I do not know the answer to it, it will take some time for me to find out the answer and get back to you. Like the other ma'am who asked me questions, like I'll get back to her after finding out answers tomorrow. So, if you can ask answers from before, then it will be better for me to address during the live sessions.
Okay, ma'am.
Yeah, we can do the same. Like we can mail you the questions
Yeah,
earlier.
yeah, you can mail me the questions. If any
Yes.
questions are there, you can mail
Uh,
me the questions. I have pinned my mail ID and I will try to address. Either I can directly address in the mail if it is possible through mail. And if you need a lot more explanation, then we can discuss in live session. That is what the live session is for actually.
yeah, actually, like on the fifth session, which you are planning about,
Yeah.
so you can give a date prior to us
Okay.
that on this we will be having doubt. So everyone, please mail me the questions. So like that we can do it. And
Okay. Then I will post an announcement, and
yes,
instead of Saturday, we will conduct it. Then like next this Saturday we are having a class. Next week Saturday also I'm having a class. So after that we can conduct it instead of Saturday. We can do it on a weekday, like Friday.
yes. So
Friday 4 to
everyone
6 we
can
can do.
message you the
Same link.
email
Yeah.
you the
I have
questions,
given
and
the link.
then
I have given my mail ID. You can mail me the questions, and if needed, I will prepare answers. If needed, I will prepare presentations also.
thank you so much for that.
Okay. Okay. Welcome. MCQ for exams. Okay. Question. How questions for exam pattern? I think yeah, it is MCQ based. Yeah. Yeah. Welcome. Any other questions? Any other queries? Mark distribution, I don't know as of yet. Old question papers are not available. Actually, I am supposed to discuss old question papers with you, but unfortunately, old question papers are not available. So I could not get any old question paper. But you do not have to worry. Questions are very easy. You go through the slides. You have any problem not understanding, like the ma'am had some problems. Time also it will be mentioned. You do not have to worry. Sufficient time will be given to you to answer questions. Questions will be relatively easy, not too much difficult. And you can answer questions on time. You will be able to do everything on time. And you just go through the slides, whatever presentation, whatever topics have been discussed, you just go through them. Everything will be covered in the presentations. Nothing will be asked outside the presentations. Like my presentations are only for your information, for your better understanding. Like carbon nanotubes was only talked about a little bit, so I elaborated a little bit more, so that in the future, if you are learning more about or working with carbon nanotubes, you know that this are other things that you have also learned. So for that, I made presentations, but my presentations are not part of your exam. It's only for your understanding, only for your own knowledge. Did all of you receive the Google Drive link with the presentation for the first week? Okay, if you have not received it, then for the next announcement, I will be sharing the link again for the Google Drive. Today, I will be uploading today's presentation as well on Google Drive, and you can download it and keep it. You can read it for your own understanding, but it is not covered in the exam. My presentations are not covered for exam. Only course, whatever is taught in your 4 weeks course, that is there for your exam. Questions will be easy. You do not have to worry too much about questions. Just read the presentations and all, you will do well. Okay. Anything else? Any other questions? Anything else you want to ask? Any other query? No other queries?
No, I don't have any queries.
Okay. Ma'am, please mail your questions to me. Tonight I will sit and
Yes,
find
ma'am.
the answers and
Yes,
tell
surely.
them to you tomorrow.
Yeah,
Okay.
thank you so much.
So thank you for joining today for the class, and it was a very nice live session. It was nice interacting with all of you. Thank you for asking very relevant questions. Even some of the questions, even I did not know the answers to. So I will try my level best to try to understand those questions and address them, and hope to see you again tomorrow at the same time from 4 to 6. So today we are ending the session a little bit early since you do not have any more questions. If you have any other questions, please, like I have already given my mail ID. You can mail me the questions. I can try to find out the answers and get back to you on time. Please give me a little bit more time. Like ma'am has asked questions, so I'll look into it and answer tomorrow. So for next session, if you have any other questions, please let me know. I will try to answer them in the other session. So, and if needed, if you really need an extra session, please let me know about that too, so that we can do the extra session also. But we will, because I have to inform IIT Madras and ask them to schedule one extra session on some other day. Okay. So thank you for joining, and I hope that we will meet again tomorrow. Bye bye. You can leave one by one.
Yeah. Bye. Thank you.
Thank you.